Thanks so much for the introduction, Beryl, and thanks everyone for joining this webinar. During today's webinar, we will go over the NovaSeq 6000 system overview and discuss the underlying chemistry and technology. Next, we will have a detailed discussion about NovaSeq reagents, as well as the sequencing workflow, which includes upstream library QC, library loading recommendations, and available analysis options for NovaSeq data. Finally, we will go over maintenance recommendations and best practices and important resources for this instrument. Let's get started with the system overview. Before diving into technical details, we will review the key features of the NovaSeq 6000. The NovaSeq 6000 uses a patterned flow cell, which was first introduced on the high-seq X and is now also used on the high-seq 4000, iSeq 100, and NextSeq 2000. The instrument utilizes the two-channel SBS chemistry, which was first introduced with the NextSeq 500 platform and is also utilized on the MiniSeq. The NovaSeq uses cartridge-based consumables, which were first introduced on the MiSeq platform. Much like all of the other platforms, the NovaSeq has a touchscreen, which makes it easy to navigate through the control software interface. For high-throughput labs, the system is ready to integrate with a LIMS system, whether that's Clarity LIMS from Illumina or a LIMS system developed from your own lab. Finally, the system has modularized hardware components, which increases the instrument uptime and serviceability. We can also connect the NovaSeq to Illumina Proactive, which helps us monitor the instrument hardware performance. For example, we will receive alerts if the instrument is operating outside of these specifications. Through the Proactive portal, Illumina Tech Support can also access run performance metrics during troubleshooting, and that helps us resolve issues quickly and potentially before they affect any sequencing runs. Now let's take a closer look at the instrument's hardware. From the top, we have a large touchscreen monitor. Directly beneath is the optical bench, which includes the optical system that is used during imaging. Moving our way down, we have the flow cell stage, which can hold 2 flow cells. Like all high-seq platforms, the NovaSeq is able to run 2 flow cells at the same time. When loading the flow cells, all you have to do is place the flow cells on the stage. The system will then automatically clamp the flow cells into place and engage the vacuum. Below the flow cell stage, we have the fluidics system. The fluidics system is separate for each side, which increases the uptime and serviceability of the instrument. The first compartment holds the cluster cartridge and the SBS cartridge and also includes a reagent chiller. The chiller is only running when the instrument is performing a sequencing run. The second compartment at the bottom contains the buffer cartridge and 2 containers for spent reagents. The larger spent reagent container is for the non-hazardous material, and the smaller one is for the formamide. For high-throughput labs, we also have the option to connect the spent reagent lines to a larger waste drum for automatic waste disposal. So now that we've taken a look at the instrument hardware, let's look into the sequencing technology. The NovaSeq uses a 2-dye chemistry, which uses 2 images per cycle to extract the intensities. This reduces the overall amount of images needed during the sequencing run and therefore reduces the runtime and the data footprint. As mentioned earlier, the NovaSeq uses cartridge-based consumables. That means that the reagents needed for clustering and sequencing are packaged into cartridges, which makes them easy to use and easy to store. The cartridges are also RFID tracked, which allows for automatic identification when loading the consumables on the instrument. The RFID also allows us to track lot numbers, consumable serial numbers, and expiration date. For example, the instrument will let you know if you accidentally loaded expired reagents or if the cycle number you chose for your sequencing run exceeds the chemistry cycles present in the cartridge. Let's move on to the flow cell technology. The NovaSeq uses a patterned flow cell. The patterned flow cell has nanowells on the top and bottom surface, which increases the data output and helps to optimize the base calling process while reducing the runtime. Each nanowell contains one cluster, and because the nanowell positions are predefined, this flow cell technology does not require template generation at the beginning of the sequencing run. The NovaSeq uses rigid registration to determine which wells contain clusters and which wells are empty. For more information on the pattern flow cell technology, click on the link on the bottom of this slide to watch a video about pattern flow cell technology. On the NovaSeq, the sample can be loaded automatically on all lanes of the flow cell, which is called the standard workflow, or for each lane separately using the Xp workflow, and more on that later. Let's look at the different NovaSeq flow cell configurations in more detail. We have the SP, the S1, and the S2 flow cell, which all have 2 lanes. And then we have the S4 flow cell, which has 4 lanes. The S2 flow cell lanes are wider as compared to the SP and S1 flow cell, which allows for higher data output. The S4 flow cell not only has more lanes, each lane is also wider as compared to SP, S1, and S2. And this gives the S4 flow cell the highest data output of up to 3,000 gigabases or 3 terabases. The output is equivalent to 25 human genomes at 30x coverage. A list of the specifications is also included here on this slide. All flow cells have a plastic casing around the edges, which allows the instrument to effectively grab the flow cell and engage the vacuum. One main advantage of having so many different data output options with the flow cells is cost-effective scalability of projects. For example, the SP and S1 flow cell have a lower data output for smaller projects or pilot studies with a faster turnaround time. And once the proof of concept of the pilot study has been established, the application can be easily scaled for production runs with S2 or S4 flow cells. So here's a comparison for those of you who currently have projects on the high-throughput 2500 in rapid run mode and how that can transition into the NovaSeq. The high-throughput S Prime kit has an output ranging from 65 to 400 gigabases depending on the read length. In comparison, the NovaSeq can generate more than double the amount of data with a shorter turnaround time. So the main takeaway here is that we're able to achieve the same level of data quality at almost half of the amount of time and double the amount of data. Let's take a closer look at the 2 sequencing workflows on the NovaSeq. We have the standard workflow and the Xp workflow. The standard workflow has a streamlined operation with less hands-on time and automated XM mixing. In this workflow, the sample will be automatically delivered after loading the instrument, and the same sample will be loaded in all lanes. In order to provide more flexibility, we also developed the Xp workflow, which allows you to sequence different library pools in different lanes. And this can be useful when you don't need a high amount of coverage per sample or if you have less sample input. The Xp workflow is very similar to the CBOT Duo workflow on the high-throughput 2500. In order to perform the Xp workflow, we will not need a CBOT, but you will need to make a one-time purchase of a flow cell loading dock. You'll also need to purchase one Xp loading kit per flow cell. This kit will come with one flow cell manifold, a set of XM reagents. And with the Xp workflow, you will manually set up the XM step. The library pools are then manually loaded onto the flow cell lanes using the loading dock and the manifold. Once the samples are loaded, the flow cell should be moved to the NovaSeq and the run should be started within 30 minutes because the XM step is time sensitive. We also have two different sequencing kits for the NovaSeq, version 1 and version 1.5. The version 1.5 reagents were launched in August 2020, and in order to run those reagents, we need to upgrade the NovaSeq control software to version 1.7. NovaSeq control software version 1.7 is compatible with both version 1 and version 1.5 reagents. With the new reagents, we have an increase of 38 SBS chemistry cycles in the cartridge. So for example, the 100 cycle kit now contains 138 cycles of SBS chemistry. To create sample sheets for those new reagents, we want to use the Illumina Experiment Manager version 1.19. We also have an S4 35 cycle kit, which is new. Further improvements include an extended minimum guaranteed shelf life from 3 to 6 months and updated Q30 score specifications. More information about version 1.5 reagents are included here on this slide. The software upgrade can be self-installed when upgrading from NovaSeq control software version 1.5 or higher. And one of the main changes includes a change in the default indexing workflow from the forward strand workflow to the reverse complement workflow with the 1.5 reagents. The reverse complement workflow uses a solution-based primer, with the index 2 sequencing primer being part of that solution. So now that we've covered a lot of information about the instrument and the instrument technology, in the next section, we're going to take a more detailed look at the available sequencing kits for the NovaSeq. With the cartridge-based technology and the RFID track consumables, the NovaSeq leverages improvements made on previous sequencers and takes high-output sequencing to the next level with less hands-on time. To perform a sequencing run on the NovaSeq, we need one NovaSeq sequencing kit. Currently, we have the following types of sequencing kit available. SP comes in 100, 200, 300, and 500 cycle options. S1 and S2 kits come in 100, 200, and 300 cycle options. And the S4 kit is available with 200 and 300 cycle options. All of these kits come with an extra 30 or 38 cycles of SBS chemistry to support dual indexing. Each sequencing kit comes with 5 items: a cluster cartridge, an SBS cartridge, a flow cell, a library tube, and the buffer cartridge. The cluster cartridge and the SBS cartridge are stored at -20 degrees and roughly the size of a MySeq cartridge or standard shoebox. The flow cell is stored at 4 degrees and about 1.5 times larger than a NextSeq flow cell. The library tube and the buffer cartridge are stored at room temperature. The buffer cartridge is about the size of a picnic basket and can weigh up to 10 pounds depending on the sequencing kit. If you're part of a very high throughput lab and need to have a constant stock of these consumables available, all of the box dimensions are listed in the site prep guide for storage space planning. So here's a more detailed look at the cluster cartridge. The cartridge is keyed in a way that it can only be loaded into the instrument in one orientation. Every other row of wells is empty. The empty wells are for the automatic post-run wash, where the buffer is pushed through the lines and into these empty wells. The reagents are used from the back to the front, and the library tube will be loaded into position 8. Position 5 to 7 are reserved for custom primers if needed for your application. Position 17 contains a 0.25% sodium hypochlorite or bleach, which is used during the post-run wash. We also have a removable position, which is number 30, for the formamide, similar to the NextSeq and MiniSeq cartridges. The SBS cartridge contains the incorporation mix, the scan mix, and the cleavage mix in the list of positions. Just like the cluster cartridge, the SBS cartridge can also contain an extra well for each reagent to catch the wash buffer during the automatic post-run wash. Similar to the cluster cartridge, the SBS cartridge can only be loaded in one orientation. One of the new features of the NovaSeq is that the scan mix is now degassed on the instrument during sequencing to help prevent bubbles during imaging. So this is what the buffer cartridge looks like. It can only be loaded into the instrument in one orientation, and it contains the buffers used during the sequencing run itself and during the post-run wash. So let's take a look at the XP components. We have 2-lane and 4-lane XP loading options to match the flow cells. As previously mentioned, you will also need a loading dock, which is just a one-time purchase and can be reused. You can also purchase 2-lane and 4-lane manifold packs that come with multiple manifolds. So now that we've learned about the NovaSeq instrument and the reagents, let's take a look at what an end-to-end workflow could look like. The NovaSeq is an open platform, and the type of application for this system is not limited. All Illumina libraries are compatible, and theoretically, any library containing the Illumina P5 and P7 adapters are compatible with this instrument. When working with custom libraries or third-party libraries, it will be up to the user to validate those libraries on the system. Let's talk about library considerations real quick. The first consideration is the loading concentration. If we're sequencing 100% Phi-X, we use a 250 picomolar loading concentration. As with all Illumina libraries and platforms, shorter libraries have a higher clustering efficiency. So depending on the library size, the library amount should be adjusted accordingly. When we sequence low diversity libraries, best practice is to start with 20% Phi-X spike-in and further optimize from there. If you're sequencing methylation libraries, a 10% Phi-X spike-in should be sufficient. Similar to other platforms, if using low-plex pooling, we do recommend that bases from both red and green channels are represented. Make sure there's at least one library that does not start with GG for the index read. If the libraries have already been diluted and denatured and transferred into the library tube, but for some reason we can't start the sequencing run, the library tube can be stored at either -20 or 4 degrees to use at a later time. However, for optimal results, we recommend to start the sequencing run right away. Library quality control is crucial for successful sequencing. As with all Illumina platforms, we recommend to use a fluorometric method for quantification. For an even more accurate measurement, consider performing a qPCR because this will only measure the libraries which cluster on the flow cell. We also recommend to confirm the quality of the library using a bioanalyzer or fragment analyzer. This confirms the library size and checks for library contaminants. Before pooling the libraries, we need to consider how much library we want to load onto the flow cell. The library input for this instrument is comparatively high because of the size of the NovaSeq flow cells. So we need to make sure we have enough sample prior to pooling. In this chart, we first have the desired final loading concentration for the flow cell in picomolar on the left, and the corresponding starting concentration of the pool on the right. For example, if we want to load the samples at 300 picomolar for onboard cluster generation, we will need to create a pool at 1.5 nanomolar. Once we made that pool, we can move forward with the denaturation and neutralization. The final loading volumes for the library tube are also listed here. Illumina also has a pooling calculator, which helps with the pooling. So you can simply enter the plexity for the pool and whether the concentrations for the individual samples have been normalized or whether they're different. Then we enter the target concentration and the total volume of the pool. The calculator will then help calculate the volumes for each sample. Just as a side note, this calculator is not particular for the NovaSeq platform and can be used for other platforms as well. So after the library has been denatured and diluted, we're now ready for sequencing. First, we need to thaw the cluster and the SBS cartridge. Ideally, we do that before the denature and dilution step because it can take a while for the cartridges to thaw completely. The cartridges tend to float in the water bath, and therefore we recommend using a wire thawing rack to help prevent them from tipping over. Once the consumables are fully thawed, confirm that there's no residual ice and invert the cartridges 10 times for mixing. Because the buffer cartridge is stored at room temperature, it can be taken directly from the storage and loaded onto the instrument. If custom primers are needed, they should be loaded into the custom primer wells or spiked into the Illumina primer wells. Refer to the custom primer guide for more information. So the next step is to remove the flow cell from the 4-degree storage, and we recommend to let it sit on room temperature for 30 minutes. This helps to prevent condensation on the glass during the sequencing run. Remove the flow cell from the plastic case and inspect both the top and the bottom for potential debris. Then, clean the glass surface and the gasket area with lint-free tissue or a Kim wipe. If for some reason the run cannot be started after the flow cell packaging has already been opened, it can be put back on 4 degrees and used within the next 24 hours. The next step is to load the denatured and diluted library pool into the library tube and insert the library tube into the cluster cartridge. We want to make sure the cap is off. We won't actually be able to load the cluster cartridge when the cap is left on. Once all of the consumables are ready, load them onto the instrument as prompted by the NovaSeq control software. And then the NovaSeq control software will register the RFID consumables and display them on the screen. Now that the run is ready to be set up, let's take a brief tour through the control software. We can select the hamburger menu in the left upper corner. From there, we can either minimize the software to access windows, we can close the control software or initiate a power cycle. We can also manage data storage and check the status of data transfer. Under the Settings menu, we can toggle between several options for data storage and analysis. The first option is manual mode. In this mode, the sequencing data will be sent directly to a network location. And due to the extremely high amount of data, we cannot save the data on the local computer. We have to send the data to a network storage location or BaseSpace. The second option is the file-based mode. If your lab has a LIMS system, this is where LIMS can be integrated with the instrument. BaseSpace Sequencing Hub is available for both manual and file-based mode with options for run monitoring only or run monitoring, storage, and analysis. Let's take a look at the process management. This is where we can monitor run processes and data transfer to BaseSpace and your network location. Before starting a new run, it is best practice to come here and verify that all processes are complete. Before starting a new run, make sure the previous runs have been transferred to the network location and BaseSpace. We can store up to 2 full S4 runs on the instrument, and the data will be stored on the Linux-based compute engine, or also called CE. Because the local computer can only store 2 runs, we have to clear that space before starting a new run. And as a side note, another cool feature of the NovaSeq is that the real-time analysis makes concatenated BCLs, which helps to reduce the overall data footprint. So now that we've taken a tour through the control software, let's walk through the run setup. As you can see, the screen is very intuitive and easy to use. Depending on how many runs we want to set up, we can select on which side we want to load the flow cell. So this can be A, B, or A and B. The next step is to follow the screen prompts for flow cell loading. Once the flow cell is loaded, load the consumables as prompted by the screen and wait for the RFID registration to complete and for the consumables to be displayed on the screen. Once the RFIDs have been read, we can now proceed with the next step. On this screen, you will be given the option to log into BaseSpace, and by entering your account credentials, you will be able to connect to BaseSpace for run monitoring and storage, or run monitoring and analysis. Next, we need to define the run parameters such as the run name, the number of cycles for each read, the network output location, and the sample sheet. The sample sheet is optional unless the run is sent to BaseSpace. If we're using BaseSpace, a sample sheet is required. For the output location on your network, we cannot use a map network drive. We can only use the UNC path to the desired network. Also, the software will not let you use the C, D, or Z drive as output location because those drives are local on the instrument. Once all of the parameters are defined, we can review the information to make sure everything is correct. Once you select Start Run, we will no longer be able to edit this information. After Start Run is selected, the instrument will perform a series of pre-run checks to make sure all of the hardware, network connections, disk space, etc. are good to go. And when all tests are passing, the run will automatically begin. If any particular checks are failing, you will be given the option to make adjustments or retry that test. So this is what it actually looks like when the run begins. On the top, we have the estimated completion date and time in case you want to plan ahead for future runs. We also display high-level metrics such as clusters passing filter, overall yield, intensity, and Q30. And at this point, you can walk away from the system and don't need to return until you're ready to set up the next run. We recommend to not touch any part of the instrument while it's running because that could lead to premature termination of the sequencing run. Just a quick overview, cluster generation takes about 3 hours. SBS chemistry takes between 4 to 6 minutes per cycle, and paradigm turnaround takes approximately 40 minutes. Once the sequencing run is completed, and if the data was streamed to BaseSpace, we have the option to generate fast queues in the cloud and then process the data in various analysis apps in BaseSpace. If you have a custom-built pipeline, the data on the network can be converted to FASTQ files using BCL to FastQ, and then port it over to your analysis pipeline for further processing. So now that we covered the individual steps, let's do a quick workflow overview. The NovaSeq 6000 is an open platform, which means that a variety of library preparation kits are supported. Sequencing requires approximately 20 minutes hands-on time, and it takes 44 hours or less to complete a sequencing run. The automatic post-run wash takes about 80 minutes. Once the sequencing run is completed, the post-run wash will automatically be performed. The wash utilizes the solution from the buffer cartridge as well as sodium hypochlorite from the cluster cartridge. As mentioned earlier, the spent reagents are separated by non-hazardous waste and formamide. We highly recommend to empty the spent reagent containers in between each run. The formamide reservoir contains about 90 milliliters of waste per run. From this volume, 10% is formamide and the other 90% is wash buffer. The cluster cartridge contains a detachable formamide position, and this is position 30 on the cluster cartridge. To remove this compartment, simply lift the white plastic on the cartridge. So finally, let's cover NovaSeq 6000 maintenance and best practices. Let's start by discussing washing and idling the system. So we already discussed that the instrument will automatically perform a post-run wash. In addition to the post-run wash, we should also perform a maintenance wash if one or more of the following circumstances are met. So if the instrument has not had a run per side for more than 14 days, if the instrument had not had a 4-lane run within the last 14 days, or if a run ended prematurely. When performing a manual maintenance wash, please be aware that the system will need a used buffer cartridge. Therefore, we recommend to save the buffer cartridges after the run is completed in case we need to perform manual washes. Cluster and SBS wash cartridges are provided with each instrument. And to perform a manual maintenance wash, we need to prepare a 0.05% tween solution that will be added to the SBS wash cartridge in the center position. We also need to prepare a 0.25% sodium hypochlorite solution that will be loaded into position 17 of the cluster wash cartridge. We highly recommend to use lab-grade sodium hypochlorite. This ensures that the starting concentration is accurate enough for our needs. Commercial sodium hypochlorite can highly vary in concentration and is not suited for NovaSeq washes. Once the tween solution and the sodium hypochlorite are prepared, we will load the wash cartridge, the used buffer cartridge, and the 4-lane wash flow cells and follow the prompts on the screen to set up the wash and start the wash. Typically, when the instrument is not in use, we recommend to simply leave it on. The reason is that there are temperature controls that we want to maintain. If we need to shut down the instrument for any reason, such as a planned power outage, we recommend to shut down the instrument through the control software. Once the screen has turned off, also toggle the switch located in the back of the instrument. When we want to turn the instrument back on, we want to toggle the switch in the back and then also press the power button located on the right side of the instrument once it starts blinking blue. And here is just a brief collection of the information from the last few slides in case you want a printout for your lab. So other best practices for the NovaSeq include that in case you encounter RFID consumable errors, we recommend to contact technical support for a further troubleshooting and assistance. We also recommend to always prepare fresh NaOH and check the pH of the NaOH stock solution regularly to make sure the pH is above 12.5. Please also always follow the recommended library quantification methods and QC steps. You can also pre-qualify the libraries prior to loading them on the NovaSeq on a smaller instrument such as the iSeq. For best results, we recommend to load the libraries quickly after denaturation to avoid secondary structures that could interfere with clustering. Last but not least, a list of important and useful resources for the NovaSeq. We have general resources such as the NovaSeq support page, the specifications page, the pooling calculator. We also have system documentation such as the SciPrep guide, the system guide, and the custom primer guide, and training videos that can help you prepare for use of the system. We also have data sets on BaseSpace available. So if you're curious to see what a NovaSeq run looks like, you can go ahead and access those data sets on BaseSpace. I would also like to highlight our support bulletins. We have a couple of bulletins that are particularly useful for this instrument. One of them helps to troubleshoot NovaSeq flow cell loading issues. Others go over how to set up and configure your output location on the instrument and how to optimize the loading concentration for the pattern flow cells for NovaSeq and also iSeq. With this, I would like to thank you for attending this webinar, and I'm happy to take questions.